Hello, my name is Samantha Rice, and I am a third-year PhD student at Oklahoma State University Center for Health Sciences under the mentorship of Dr. Randy Weimar, and my poster is Molecular Analysis of the Systemic Dermatoses of Morgellons disease. Morgellons disease is a multi-system infectious disease historically viewed as controversial and poorly understood by the medical community. These contentions involve difficulties in diagnosis, as symptoms of MD have similarities consistent with a psychiatric disorder involving the false belief of infestation by parasites, also known as delusional parasitosis or delusional infestation. Currently, the factors determining the etiology and transmission of MD are still unknown, and the dispute surrounding Morgellons is substantial. MD doesn't discriminate. It affects people of all ages, genders, and ethnicities. In MD patients, a distinct feature of near microscopic and microscopic fibers is visual within the dermopathy, possessing unique characterization and formations within the cutaneous and subcutaneous layers of the skin. These unique fibers are spontaneous and consist of a multitude of colors. Although the colors of fiber is not fully understood, fiber-like filaments are perceived to be caused by an overproduction of keratin and collagen, with blue filaments containing granules of melanin. However, analysis from past research has given mixed results from unknown to keratin. Other signs and symptoms of MD include crawling sensation on and under the skin, intense itching, severe fatigue, cognitive difficulties, and behavioral effects. The aim of the study is to investigate if an infectious etiology of the systemic dermatoses is present. Previous research endeavors suggest MD is a disorder associated with a tick-borne illness caused by the bacterium Borrelia burgdorferi, a pathogenic spirochete and the causative agent of Lyme disease transmitted to humans through the bite of an infected black-legged tick, Ixodes scapularis. MD studies are currently focused on spirochetes as a causative agent to understand if a co-infection exists and/or multifactorial etiology is a cohort. We hypothesize the presence of multiple pathogenic organisms found in dermatological specimens, suggesting infectious pathogens in MD etiology. Recognition of potential coexistence of multiple pathogens in MD etiology may stimulate the development of novel approaches to diagnosis and treatment, therefore allowing multiple diagnostic approaches to be applied simultaneously to detect for major pathogens in MD. In our lab, epithelial tissue samples are collected and de-identified for DNA extraction, and polymerase chain reaction, also known as PCR, is performed using specially designed primers to detect if unusual microbial organisms are present. These bacteria are Bartonella henselae, the causative agent for cat scratch fever, Borrelia burgdorferi, the causative agent for Lyme disease, Helicobacter pylori, the bacterium that causes stomach ulcers, and Treponema denticola, the bacterium that causes periodontal disease. After amplification, PCR products are then visualized on a 1 to 2% agarose gel, stained with CyberSafe, and visualized on the gel doc imager. Positive bands are excised and purified from gels, and the eluted DNA is quantified using a nanodrop spectrophotometer. The DNA is then sent to OSU's core facility for Sanger sequencing with bioinformatic tools implemented upon completion. Nucleotide sequences are interpreted for verification of our genes of interest. Our results indicate the presence of multiple pathogenic organisms found in dermatological specimen, suggesting an infectious etiology of the dermopathy is present, supporting our hypothesis. DNA analysis of Bartonella henselae, Helicobacter pylori, Borrelia burgdorferi, and Treponema denticola have been successfully matched with GenBank, the National Institutes of Health's genetic sequencing database. Environmental contaminant controls were implemented to demonstrate these bacterial agents are not readily found in the environment, that include epithelial tissue samples and surfaces in random settings. Replication of preliminary data is ongoing for the credibility of scientific claims. Future research needs to be conducted to continue the exploration of etiological causes to support our findings, with additional primers and controls being assayed in future samples. Through further analysis, the potential coexistence of multiple pathogens in MD may hold the answers to this unsolved mystery and bring a better understanding for MD to be recognized in the medical community. Thank you for taking the time to listen to my presentation, and I hope you have a wonderful day.